AP Biology, the chemistry of life, water and macromolecules, review for the test. These are some terms that are in Chapter 3, dealing with water. Polar covalent bonds. The source of all of water's amazing properties can be traced back to the water's ability to make polar covalent bonds between oxygen and hydrogen. Within the water molecule, you have a strong covalent bond, a sharing of a pair of electrons between hydrogen and oxygen that is not shared equally. Oxygen acts as kind of an electron hog. It's very electronegative. As the electrons spend more time at the oxygen, the side with the oxygen has a slight negative charge. The hydrogen has one proton and one electron. Since the electrons are being drawn to the oxygen, the proton left behind gives the hydrogen side of the water molecule a slight positive charge. Now, this is not a full charge like an ion. It's a partial charge represented by the symbol that looks kind of like an 8. As a result of water's polarity, or unequal sharing of electrons between oxygen and hydrogen, you have a negative side, or negative pole, and a positive side, or positive pole. This leads to many other properties of water. Hydrogen bonds. As a result of the polar covalent bond within the water molecule, the unequal sharing of electrons between oxygen and hydrogen, you have a positive side and a negative side of the water. This is going to cause an attraction for other water molecules as well as other molecules that have polarity or ionic properties. Here we have the hydrogen side of one water molecule attracted to the oxygen of a neighboring water molecule. The weak attraction between the two water molecules is called a hydrogen bond. This is very easy to break. However, as a result of hydrogen bonding in water, water has a couple of other properties that you should be aware of. Cohesion. Hydrogen bonding between water molecules is called cohesion. Water is sticky, or hydrogen bonds, to itself. This creates things like surface tension, which allows small, light objects like insects to walk on water. Also, if you tried to suck up sugar through a straw, since the sugar that is far away from your mouth doesn't really bond to the sugar that you're actually taking into your mouth, they don't stick to each other, and the sugar just kind of falls back down. However, because water is sticky to itself, the middle water that's in a straw kind of holds on to each other, and as more water comes into your mouth, the other water at the bottom of your straw gets brought up. Adhesion is water hydrogen bonding to other substances that are also ionic or polar. For example, in glass tubes, you have something called capillary action, or any small tube that's attracted to water. The water is hydrogen bonding to the glass in, say, a capillary tube, and as it kind of pulls itself up by hydrogen bonding, that adhesion will draw up the water up the tube. The cohesion of the middle water not touching the glass keeps the water in the middle from falling back down again. A meniscus is what's read on a graduated cylinder. The low part of the meniscus is just the pull of gravity trying to pull the water down. However, the water kind of creeps up the side of the graduated cylinder, and the reason why is because of the hydrogen bonding or adhesion. Now, the reason why it doesn't go up very far is just because there's just so much mass of water in the middle of a graduated cylinder that the hydrogen bonding on the sides doesn't have enough strength to bring it up very far. Really, you can only bring up water in a very small tube, like in the xylem tissue or tubes of a plant, in order to bring up water to the leaves. Also, water can climb up fiber. If you ever noticed when you throw a paper towel on a wet spill, the water just kind of creeps along the paper towel until the entire paper towel is wet. That's all hydrogen bonding. Another term for that hydrogen bonding to another substance, the paper towel's cellulose, is adhesion. Remember that paper comes from trees, and trees are mainly made of a macromolecule called cellulose, which is composed of simple sugars like glucose. Glucose, C6H12O6, has a fair amount of oxygen, and that oxygen in the glucose is going to be attracted to the water. Hydrophobic means repelled by the water, and typically things with a lot of carbon-hydrogen bonds, which are covalent bonds, but since it's equal sharing of electrons, there's no charges built up on these fatty acid tails in this triglyceride. So, as a result, no charges, nothing for water to adhere to, it's hydrophobic. This is a triglyceride. It has 3 fatty acid tails and 1 glycerol molecule made by dehydration synthesis. This is the general form of fats and oils. Water is the solvent of life, and that's probably the most important property of water. It's able to dissolve other polar or ionic compounds that we need for keeping ourselves alive. Since water dissolves these things by surrounding it completely, it's able to be transported throughout the body in fluid like our blood. Even if it's a organism without blood, something like a tree which has sap, you can still dissolve important life molecules within that water and transport it from point A to point B. As a result, water is very important as the universal solvent, and especially for dissolving life's molecules. Even some proteins, represented by the purple on the right-hand side, have hydrophilic R groups that can be attracted to the water and dissolve within the water, depending on the protein. Hydrophilic means loving water or having affinity toward water. These are compounds that are either polar or ionic. Why is ice floating important? As a result of the hydrogen bonding, the water molecules kind of set themselves up in a crystalline pattern. As a result, they become less dense as a solid instead of more dense like most substances when they go from a liquid to a solid. Because ice is less dense and floats on top of water, it insulates the water below even if the air temperature above is much colder. This will keep the water below the ice liquid, because the ice above kind of acts as like a blanket insulating the water below. And life in the lake or a pond, for example, fish or algae, don't necessarily die in the winter. If ice sank, all the ponds, lakes, and even oceans would freeze solid from the bottom up. It's very cold at the bottom of the ocean, and in summer only the top few inches would thaw. So it's kind of a good thing that ice floats. Specific heat is the amount of energy required to raise the temperature of a substance 1 degree Celsius. Water has a high specific heat due to the hydrogen bonding. You have to break the hydrogen bonds first before you can raise its temperature. So that means that water resists temperature changes and takes a lot of heat energy to heat it up or cool it down. This moderates not only the temperatures on the planet, but also within our bodies as well, preventing rapid changes in temperature as a result of all that water. That's one reason why people like to live in places like California. All that water from the Pacific Ocean acts to moderate the temperatures on the land. As you might guess, places with very little water, deserts, middle of continents, grasslands, have more extreme temperature changes as a result of water not being there, trapping heat and releasing it slowly, either during the winter or during the night, if it's the case of a desert. Evaporative cooling. As the water molecules heat up, the faster ones break free of their hydrogen bonds and leave, leaving the cooler ones behind, and that cools down our bodies. That's called evaporative cooling, as a result of water's high heat of vaporization, or a lot of energy to break those hydrogen bonds in order to turn it into a gas. So here's a simple concept map. Basically, all of water's properties can be traced down to water's polarity, the unequal sharing of electrons within the water molecule. That leads to hydrogen bonding between water and water to other substances. And it takes a lot of energy to break those hydrogen bonds or raise or lower its temperature as a result. Also forms a crystal lattice when it freezes, that's cohesion. Clings to the sides of tubes and trees. Adhesion to the sides of the tubes in the trees, that's going to be to the cellulose. Cohesion prevents the water in the middle of the tubes in the trees from falling back down, and evaporation from the leaves allows more water from the roots to be brought up. The most important property of water is that it dissolves other substances, and that's adhesion basically, and can be used to transport things around the body. Basically, all of life's chemical reactions happen within water. pH scale. pH scale is from 1 to 14, and 7 is in the middle. That's neutral. 1 in 554 million water molecules is dissociated into hydrogen and hydroxide ions and acts as a weak buffer. What that means is that if it's a base or has excess hydroxide ions, some of that water, very little of that water, will bind to the hydrogen ions in that water, will bind to the hydroxide, neutralizing it and resisting the change in pH. The reverse is true as well. If you have an acid solution or something with an excess of hydrogen ions, because water forms hydroxide ions, some of the hydroxide binds to the hydrogen ions, neutralizing them, turning into water, and neutralizes that acid. Again, resisting the acid changes in an aqueous solution. This is how water acts as a weak buffer. It doesn't do a great job of it, but it does help out a little bit. That's kind of important since you have water in your blood, and that's going to resist some acid or alkaline changes to your blood's chemistry. The pH scale is from 0 to 14, and in the middle we have 7. Acids are under 7, bases are above 7. Macromolecules. Here are some key terms for the unit on macromolecules. Chemistry of life. Remember that organic chemistry is the study of carbon compounds. The big thing that you need to know about carbon chemistry is that it forms 4 stable covalent bonds and can form bonds with itself. This leads to an incredible amount of diversity in the amount of molecules that carbon can make. Organic chemistry refers to carbon being at the backbone or the starting point for adding functional groups onto later and forming the macromolecules that compose living tissues. Why study carbon? Well, all living things are made of cells, and again, carbon compounds, about 25% of life is made up of these carbon compounds of carbohydrates, lipids, proteins, and nucleic acids, together known as the macromolecules, though the least macro of the bunch are the lipids. Remember that living things are about 72% water and 3% salts. Water, of course, used as a life solvent as well as other reasons. Salts are used for things like the nervous system and conducting nerve impulses. Some salts, like calcium salts, are found in our bones. Isomers. Remember that an isomer is a molecule with the same molecular formula but different structures. Like you could have a carbon coming off the middle carbon with other functional groups. Typically, the left-handed version of a molecule is the one that's active. The right-handed version is not active in biological systems. As far as what that means exactly, you'll have to save that for your organic chemistry teacher's class. Macromolecule. Smaller organic molecules join together to form larger molecules. These are the macromolecules we're going to talk about now: carbohydrates, lipids, proteins, and nucleic acids. So the first one we're going to deal with is carbohydrates. How to build a polymer. Remember, this is called dehydration synthesis. We pull out water to bring the monomers together to make the polymer bigger. Remember, this requires adding energy. It's going to be endergonic and anabolic and requires the use of enzymes. Hydrolysis is breaking apart the molecule into smaller monomers. You can break down the word. Hydro refers to water in this case. However, it could also refer to hydrogen in other cases. Lysis means to break apart, so hydrolysis literally means to break apart using water. In the previous example for dehydration synthesis, dehydration refers to removing water. Synthesis means to put together. So dehydration synthesis literally means to put together by removing water. An example of this would be like building up muscle tissue. The actin and myosin has to be made, those are proteins, by linking together amino acids. When you make muscle, linking together the amino acids would be an example of dehydration synthesis. The ribosome does this in order to put together the amino acids to make a protein. An example of hydrolysis would be digestion. When you break down proteins, let's say a chicken nugget, then you're going to use enzymes and release energy and use up water, or you need to add water to the molecule in order to make this reaction happen. Think of it as an opposite or reverse of dehydration synthesis. All right, the first carbon compound is carbohydrates. Monomers are sugars like glucose that are used directly as energy. Polymers include starch, glycogen, cellulose, and chitin, which store energy or is used as structures. Carbohydrates have 3 elements in their molecular structure: carbon, hydrogen, and oxygen in a 1 to 2 to 1 ratio. For example, glucose, C6H12O6, has a 1 carbon for every 2 hydrogen for every 1 oxygen. That is a carbohydrate. They don't always have this exact ratio of 1 to 2 to 1, especially for the polysaccharides, because some of the branching reduces the number of hydrogen or other elements. The function of carbohydrates are energy. Primarily glucose is going to be directly used as energy in the process of cell respiration to make ATP. Energy storage, the 2 energy storage molecules are glycogen and amylose, also called plant starch. Raw materials like glucose can be used to string together and make structural materials like cellulose. Cellulose is the main cell wall component of plants. Also, structural materials like chitin are made of glucose, modified glucose, that will make up the structural cell walls of fungi. The monomers are sugars. Specifically, glucose is the most common monomer. Monosaccharides are 1 sugars. The most important one is glucose, but it also includes galactose and fructose. Disaccharide, or 2 sugars, there's only one that we want to talk about, and that's sucrose. That's just one glucose and fructose chemically bonded together by a glycosidic linkage. Remember, glycosidic linkage is the special name we give to the covalent bond between two sugars. Polysaccharides, or many sugars, they're large polymers. An example would be starch, and they tend to be very, very macro indeed. This is glucose over here. Remember that every angle here is a carbon. Since in organic chemistry carbon is so common, we don't even write in the carbons to make it easier. Polysaccharides cost very little energy to build, and they're very easy to reverse and get that energy out of it. So when you need quick energy, you want a polysaccharide. If you are in some kind of sports event and you needed energy, you wouldn't want to go with a fat or a protein. You'd want to have some kind of polysaccharide like starch in order to get that energy quickly. If you had a long running event, then maybe you'd want to have some proteins for slower release of energy. The function of polysaccharides is energy storage, starch in plants, also called amylose, glycogen, also called animal starch in animals. They're also used as building materials, cellulose in plants, and chitin in arthropods and fungi. Chitin is the modified sugar chains, that's a polysaccharide, many sugars, that makes up the exoskeleton of arthropods like insects. It's interesting to think that insect exoskeletons is actually made of sugar. Here we have some examples of polysaccharides. Plant starch, also called amylose. The little green hexagon represents glucose, chained together by covalent bonds. That's not very branched. Examples of things with lots of starch would be rice, potatoes, noodles. Animal starch, also called glycogen, is very similar to plant starch, except it's more highly branched. We store this in our muscle and liver for quick energy. Remember that starch is easy to break down by most animals. We have the enzyme to attack this alpha linkage for this glucose monomer. So, for starch, we can easily break this down. However, cellulose we cannot break down. There's an isomer, or a version of glucose that's made by plants that creates a bond called a beta linkage that we can't break down. We don't have an enzyme for that. Since we have no enzyme that fits this bond, we can't break down cellulose, and it passes through our body unchanged. Now, some organisms do have that ability to break down cellulose, but they're not animals. Some things that live inside animals, like the protists that live inside cows and termites, are able to break down that cellulose into sugars, and then the animal that uses it gets an advantage. That would be a good example of mutualism. Cellulose is the most abundant organic compound on the earth, just because there's just so many producers. Remember the biomass pyramid, and all those producers have cell walls made of cellulose. Now we're moving on to lipids. They don't form monomers. Includes fats, oils, and phospholipids, as well as steroids. Functions are twice as much energy as carbohydrates, cushion organs, insulation, and making cell membranes. Fats are long hydrocarbon chains. They're nonpolar and hydrophobic, so they don't dissolve in water. They're not attracted to water, so you can't dissolve them. The function is energy storage, twice as much energy as carbohydrates or proteins, cushioning organs, and insulate body. Just talked about that. Saturated fats, remember, there is no double bonds in a saturated fat. Think of it as saturated or filled up with as many hydrogens as you can put in there. As a result, they form long straight chains that lie flat on top of each other that forms a solid at room temperature. They get really compact. Animals form saturated fats, but you can make a unsaturated fat saturated by adding extra hydrogen or hydrogenating the fat or oil. Unfortunately, saturated fats contribute to arteriosclerosis, or plaque deposits that build up in your arteries that increase your blood pressure. And over a lifetime of increased blood pressure, a bursted artery is a bad day. That's called an aneurysm. If it's a burst artery to the brain, that would be called something like a stroke. Unsaturated fats have a lot of double bonds, and plants and fish end up making this. Fats and oils are very similar to each other. The only difference really is the double bonds that creates a liquid instead of a solid at room temperature. Vegetable oils are liquid. They have lots of double bonds. The reason why is the double bond between the carbons causes a kink in the fatty acid tail, and the molecule doesn't lie, the other molecules don't lie flat on top of these triglycerides, and as a result, they slide past each other. That creates an oil or a liquid. Unsaturated fats tend to be more healthy than saturated fats. If you had more than one double bond, we would call that polyunsaturated because there's more than one or poly double bonds. Phospholipids are very important to living things. They have a dual personality, unlike the fats and oils that are all hydrophobic. The phosphate group at the top has a lot of oxygen that's attracted to the water. That part's attracted to the water, or hydrophilic. The fatty acid tails are all carbon-hydrogen and are not attracted to the water. That makes them hydrophobic. Why is that important? Phospholipids make up our cell membranes. They self-arrange into two layers called a bilayer, or two layers of phospholipids, where the fatty acid tails try to escape the water on the inside because they're hydrophobic, carbon-hydrogen, and the hydrophilic heads, that are the phosphate groups, are attracted to the water. They're attracted to the water both outside the cell, represented over here on top, and inside the cell, represented by the yellow at the bottom. Remember that phospholipids make up our cell membrane. If you can connect that back to our phosphorus cycle, remember that's one of the reasons why plants need phosphorus in order to make phospholipid membranes, which is required if you're going to make more cells. Steroids have a 4-ring structure, and all steroids start off as cholesterol. Cholesterol is just modified by enzymes, snipping off functional groups, adding others, to make the sex hormones. Now, it's only the sex hormones that are lipids. The other hormones typically are not lipids. For example, insulin is not a sex hormone. It is a protein hormone used for sugar homeostasis. Here we have some diversity in the sex hormones, the steroids. We have the estrogens and androgens, testosterone, male hormones, and depending on which functional groups you get on this sex hormone will determine how it acts in the body with different targets, fitting different receptor sites, and turn on the metabolic pathways to make you a male or a female. That's a lot of difference from female and male, just as a result of having the different functional groups on cholesterol, or a modified cholesterol, really. So remember, cholesterol is the starting point, and then you modify it to make your sex hormones, whether it's female or male sex hormones. Cholesterol is an important part of cell membranes in animals. They increase the fluidity, allow the phospholipids to slide past each other more easily. It's also the starting point or precursor of the steroids, and high levels in the blood contribute to cardiovascular disease and plaque deposits, which increase blood pressure. Nucleic acids. Nucleic acids function to store and transmit hereditary information. Examples are RNA and DNA. Structure, monomers, or the building blocks, are the nucleotides that are strung together to make the DNA or RNA. Remember, nucleotides have 3 parts. They're fairly complicated for a monomer. There is a phosphate and sugar on the backbone, which are connected together, sugar phosphate, sugar phosphate, sugar phosphate, by dehydration synthesis, creating a covalent bond called a phosphodiester linkage. The nitrogenous base in DNA could be adenine, guanine, cytosine, or thymine, and they hang off the sugar phosphate backbone. All of this is held together by covalent bonds. However, between the two sides of DNA, we're going to be holding the nitrogenous bases together by only hydrogen bonds, a weak bond. Remember that RNA gets ribose, DNA gets deoxyribose as the sugar. They're just a slightly different type of sugar. It doesn't taste sweet because once you chemically combine it to other things, you change their properties, and it doesn't interact with your nervous system to make it taste sweet. The double helix. DNA forms a double helix, basically looks like a winding staircase, where the sugar phosphate is going to be represented by the blue. That's going to make up the sides or the backbone. Not really a backbone, just a kind of an attachment point for other things, and in this case, nitrogenous bases. The nitrogenous bases hang off the sugar phosphate backbone. A, capital letter, straight line, always binds with capital letter T, straight lines. Curvy letter C always binds, or complementary pairs, with G, also curvy letters. So complementary base pairing rules: straight lines with straight lines, A's with T's, curvy with curvy, C with G's. Remember that hydrogen bonds are important because we need to be able to read those codes in order to do transcription and translation, and we need to make copies of the DNA during replication, which means that we're going to have to break the two sides of that DNA molecule in order to add new nucleotides during semi-conservative replication, where one old side is conserved and one new side is made in the new DNA molecule. Replication, the two sides of the DNA double helix are complementary. If you have one side, you can build the other. So when these two sides split apart during replication, it's very easy to make the new strand of DNA because of the complementary base pairing of the two sides. Enzymes bring in the correct nucleotides and nitrogenous bases to pair up on both sides of the new DNA molecule, and now you have two new DNA from one DNA. We learned about DNA structure from Watson and Crick, the three-dimensional shape. Basically here we have replication, new strand, complementary to the old strand in semi-conservative replication. Proteins, monomers are the amino acids. Polymers include polypeptides, which have a complex three-dimensional structure. They're very, very diverse as far as their role in living systems. The most important role of proteins would be as enzymes. Enzymes bring things together or break things apart during chemical reactions by lowering activation energy. Other roles for proteins include things like structure. We make our hair out of keratin. Collagen is a connective tissue. Carriers and transport, basically to let things in and out of cells. We have protein channels to let things in and out. Receptors and binding. Defense proteins are called antibodies, which help clump up invaders to make the immune system more efficient. Contraction proteins are actin and myosin. Actin and myosin are the two proteins that make up your muscle in order for you to move. Basically, actin and myosin slide past each other in order for muscles to contract. Signaling. Other than the sex hormones, many hormones are protein hormones, like insulin, that are used to communicate and maintain homeostasis in the body. And we can store proteins. For example, bean seeds will store proteins for later use. The monomers of proteins are amino acids. There's 20 different amino acids. What determines which amino acid is which is not the NCC backbone, but what hangs off the middle carbon, called an R group. R group refers to the sequence of elements or atoms that will define that amino acid as being what it is. The R groups will also have important interaction with other R groups when this protein folds into its final tertiary or three-dimensional shape. Remember, we're doing this, we're putting together amino acids by dehydration synthesis, pulling out of water to bring together a larger polypeptide chain. The peptide bond is the special name for the covalent bond between amino acids. Polypeptide is synonymous with protein sometimes, but it's not really the same. Polypeptide just really refers to the string of amino acids chained together. It's not really a protein until it folds into its complex three-dimensional shape. Here we have another example. The N terminal will be the starting point, and the C terminal will be the ending point. So it goes from NCC, 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 all the way down the line. That's what the ribosome's doing, chaining together amino acids based on that messenger RNA code at the ribosome. As you can see, the different R groups coming off the middle carbon will define these amino acids as different from each other, one of the 20 of the amino acids that are available to living things. Proteins form a complex three-dimensional shape that will determine its function. It will fit other molecular substrates in order to act on them and catalyze chemical reactions. Also, protein structure will be involved with the other roles of proteins as well. If it's an antibody, the shape of that antibody will affect what it can bind to and defend your body against. Here we have a review of how proteins fold. The primary structure is just the amino acid sequence determined by DNA, held together by tough bonds called covalent bonds. Special name for it is peptide bonds in proteins. I remember P in peptide, P in proteins. In nucleic acids, it also started with a P, phosphodiester linkages between the nucleotides. However, I remember that one by the phosphate backbone. That's how I remember phosphodiester linkages for nucleotide to nucleotide. The secondary structure is only going to be caused by the hydrogen bonds within the R groups, and that's going to form two structures, either an alpha helix or a beta pleated sheet. Then the final three-dimensional shape of the protein will be determined by the interactions of the R groups with hydrophobic and hydrophilic interactions. Hydrophobic R groups with lots of carbon and hydrogen will try to fold away from the water, while hydrophilic R groups will try to be attracted to the water, which will help fold it. Hydrogen bonding between R groups will help fold the protein, as well as ionic bonds between things like ammonia, and disulfide bridges, or two sulfurs that will come together to help fold the protein. Now, some proteins will form a quaternary structure, but not all. An example would be collagen and hemoglobin. Hemoglobin is composed of 4 different polypeptides in order for the protein to be functional, held together by hydrogen bonds. Here we have an example of the quaternary structure, hemoglobin having 4 different polypeptides coming together to form its final structure. Collagen has 3 polypeptides to form its final structure. Collagen is used in connective tissue under the skin, as well as tendons holding muscle to bone. Chaperonin proteins will help proteins to fold into its final 3-dimensional shape by the interactions with the chaperonin proteins R groups to kind of twist this thing into its final tertiary structure. Denaturing a protein or losing, disrupting its 3-dimensional shape or tertiary structure. pH, hydrogen ions and hydroxide ions can affect the ionic bonds as well as other bonds within the protein and disrupt its 3-dimensional shape. Salt, positive charges from sodium or negative charges from chlorine or other salts can disrupt ionic bonding and other charges within the protein and misfold it. Temperature, heat is basically just particle kinetic energy. When you heat something up, the particles move faster. If they move so fast that the hydrogen bonds can't hold together, you break your hydrogen bonds, the tertiary structure no longer is maintained, and the protein is denatured. An example would be eggs. When you cook eggs, well, you start off with liquid eggs, but as you heat them up, you denature or disrupt the tertiary structure of the egg proteins, basically disrupting the hydrogen bonding, and then eventually it loses its shape. Now, you can't uncook an egg. However, some proteins can return back to their functional shape. This is also one reason why it's dangerous to get a fever. If your fever is too high, you denature the proteins in your body that carry out life processes, and without the proteins that carry out life processes, the enzymes working, you're not going to stay alive very long. It's very important that if you have a fever, that you try to get cooled down very quickly. After 105 degrees Fahrenheit, that could be life-threatening. Enzymes and ATP. Here are some key terms for Chapter 6, Enzymes and ATP. Remember, metabolism is building up or breaking down bonds. We have some new words here. Atabolic, or really anabolic, means adding monomers to form a larger molecule. Catabolic, think catabolic, or cutting up a molecule into smaller molecules. Remember, catabolic reactions are exergonic, or energy releasing. Dehydration synthesis and anabolic reactions are energy storing. Here we have 2 graphs representing exergonic and endergonic reactions. For example, when you have a lit match, you have to add energy to the match before you can destabilize the bonds within the wood to start on fire. So imagine, you add energy, you strike the match on a matchbook, and eventually enough energy is added where energy is now released. You have a chemical reaction occurring, which is the fire, and you're now releasing not only the energy that you added to get the whole reaction started, but you have extra energy being released. That's going to be the fire that's given off a lot of energy. This energy that's released beyond the activation energy, or the energy to get the reaction started, is called free energy. And exergonic reactions release free energy, the energy that can be used to do work. Endergonic reactions store energy. So when you're building up muscle tissue, when you're making actin and myosin proteins, you're chaining those amino acids together. That requires energy from your body. Then the products, or the proteins being made, actin and myosin, have stored energy, but they're not going to release energy. You're storing the energy between the amino acids. Enzymes are biological catalysts. Catalysts speed up the chemical reactions by lowering the activation energy. If you take a look over here, for example, with a match, you have to add a lot of energy to that match before it starts on fire. In biological systems, fire would destroy us. We have something that lowers the amount of energy to get a chemical reaction started. Anything that lowers a chemical reaction's activation energy is called a catalyst. However, biological catalysts are called enzymes. There are some other ones called ribozymes that we'll talk about in a future class, but enzymes are by far the most common. Enzymes lower activation energy so that chemical reactions can get started without a large amount of energy input or a lot of temperature required. They facilitate chemical reactions and increase the rate of reaction without being consumed up themselves. As you can see with this purple enzyme at the bottom, it's not getting used up, even though it's breaking down this molecule here, probably sucrose, into glucose and fructose. They're required for most biological reactions. You do need to know all this. And they're highly specific. Each enzyme only acts on one substrate or one thing that it's going to put together or break apart. They're also used to control chemical reactions within living systems as well. Enzymes are very important to biology. Lock and key model. It's fairly simplistic. However, it's the 3D structure of the enzyme that fits what it acts on. The molecule that's being acted upon by an enzyme is called a substrate. So that's a new word there. Substrate acted upon by an enzyme. Here we have an example. Over here you see all those little dots representing the amino acids with all their little R groups interacting to fold this protein enzyme into its final three-dimensional shape. As a result of this complex three-dimensional shape, you have one little spot on this enzyme that will fit the substrate. This little spot here that fits the substrate is called the active site of the enzyme. Here we have the enzyme called sucrase breaking down a molecule called sucrose or disaccharide. Enzymes are typically named with an ACE ending, so it's fairly easy to know if you're dealing with an enzyme. Sucrase acts on sucrose. However, just because you know it's an enzyme doesn't necessarily tell you whether it's a catabolic or anabolic enzyme or an enzyme that puts things together or breaks things apart. Here we have a catabolic enzyme or an enzyme that breaks things apart. The substrate or sucrose will fit into the active site. You'll have a shape change as a result of the interactions with the active site and lower activation energy, allowing the covalent bond between the glucose and the fructose to break and releasing the glucose and fructose in the small intestine, if this is where we're breaking it down. The shape of this protein, very important. If this ever becomes denatured, then the active site will no longer fit the sucrose and the protein will not be able to do its thing. Now here's a more realistic model of how enzymes work. Basically, once you have the substrate interact with the R groups of the active site, the whole protein starts to kind of change its shape a little bit, and as a result will either bring things together to cause a chemical reaction to occur in anabolism, or building up a molecule, or in the case of other enzymes, the substrate, maybe two substrates, bind to the active site and pull further apart. It doesn't always have to come closer together. If it pulls further apart, then it might weaken the bond and allow catabolism to occur, or breaking down a molecule into simpler molecules. Some factors that affect enzymes: enzyme concentration, substrate concentration, temperature, pH, salinity, activators, and inhibitors.